Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Well, we finally made it to part 5 in my final video on sexual reproduction and inheritance. So, the 3 things I'm going to discuss today are Thomas Hunt Morgan and why he, in my mind, is the father of modern genetics. Then I'll talk about the extensions to Mendel because, Mendelian genetics is very limited. And then I'll finish up with some common misconceptions about genetics. Gregor Mendel has stolen the spotlight when it comes to being the father of modern genetics. And like I said, deservedly so. But when it comes to me and who I think of as the real father of modern genetics, it's really Thomas Hunt Morgan studying fruit flies. He discovered the chromosomal basis of inheritance. He discovered linked genes crossing over and mutations. Isn't that awesome? So he brought in this age of modern genetics. And he did this by studying fruit flies. And to this day, over 100 years later, we still use the fruit fly as a model organism to understand genetics. It's amazing. So he discovered mutations, and he discovered it in fruit flies, of course. And if you notice on the bottom, that fruit fly has a bright red eye. That's the wild type. That's what they normally look like. And then the one above it has a white eye. That is a mutation. And you realize that mutations are random and spontaneous. They can just change at any time. So they actually tried to induce mutations on these fruit flies to see what would show up. And then they did breeding experiments. So mutations are important because that's your ultimate source of variation. So for example, whenever you're creating new alleles, you need a mutation to do that. So when it comes to the pea flowers, white is a new allele compared to purple. And because white can attract pollinators, it's not a bad allele, it's not deleterious. It can bring in different types of pollinators, different types of insects. And in humans, O blood is also a mutation from A blood. He also discovered linked genes. Think about it this way. We have 23 pairs of chromosomes, and we have over 20,000 genes. There's no way that all genes can be inherited independently of each other. Most share some space on a chromosome, so that we would discover they are linked. So this is not following strict Mendelian genetics. And he was actually able to figure out how closely genes are to each other based on enormous breeding experiments. Because the closer they are together, the more likely genes are inherited together. The further apart they are on the chromosome, the more likely that crossing over will break them up. So wow, he discovered linked genes and crossing over and mutations? That's amazing. So he says, if they're on different chromosomes, then they do follow Mendel and they are not linked. And most genes, they just don't strict Mendelian inheritance. Thomas Hunt Morgan's greatly expanding on Gregor Mendel, and then rather than identifying the pattern, he started to understand what was causing those patterns of inheritance. How are these things being inherited? And looking at the molecular basis of it, because that's where we're at today. And he also showed that, genetics is quite complicated. Mendel, he came up with these rules of inheritance, like one gene, two alleles, but most genes don't follow those rules. So think about eye color. I've got all of these different colors of eyes. Eye color is quite complicated. There might be as many as 15 different genes involved with eye color, and they're acting together to create unique eye colors in people. So we have a lot of extensions to Mendelian genetics. And like I said, Thomas Hunt Morgan was the first to show how complicated genetics is. One of them is you don't have to have just two alleles. You can have one allele. You could have many alleles. Great example, A, B, and O blood type. Those are 3 different alleles. Now, in a human, so at any one time, you can only have 2 alleles, right? Because we only have 2 copies of our chromosomes. Even though in humans, we talk about A, B, and O, there might be like 30 different versions of those throughout the human population, variations on A's and B's. But no matter what, you only get 2, but in a population, you can have multiple alleles. And you can also be codominant. Anybody out there AB blood? Well, if you're A and B blood, what that means is you're expressing the A antigen and the B antigen. You're expressing both of them. So that's codominance. One's not dominant over the other. O, on the other hand, is a recessive allele. You have to have 2 copies of O to be O-type blood. And in the case of O is a mutated A. You don't produce the A, so you come out not having a surface antigen. You can also have incomplete dominance. Now, incomplete dominance is a little tricky here because you may only be producing a little bit of something that at the surface looks blended. But one of them is sickle cell anemia or sickle cell disease. Sickle cell disease is caused by a single mutation. And what it does is it causes the blood to be sickle shaped if you're homozygous for it. If you're heterozygous, your blood aren't quite circular, they're a little oval, and that gives some protection to malaria. Now, if you've watched my videos before, you know I've probably picked on mosquitoes, at least certain species of mosquitoes, because they are the most dangerous animal on the planet, not because they're going to eat you, but because they spread diseases like malaria. And malaria kills more people than any other single disease on the planet. So any protection against malaria is a good thing. So sickle cell disease, the heterozygous form, like I said, is in between the normal and the full-blown disease. Another example of incomplete dominance, and this looks kind of blended, and that's only because you're producing half the product. This one is for hypercholesterolemia. Okay, hyper, I got it above, right? Cholesterol. What this is a condition where you have high cholesterol levels. You see, your body makes cholesterol and then can ship it throughout your body. And we put it in these lipoproteins to carry it out. Well, for your cells to receive the lipoproteins, you have to have receptors on there. And if you have hypocholesterolemia, you have a mutant version that does not produce those receptors. So if you're heterozygous, you produce half the receptors. Your blood cholesterol is high. You will likely die in your 40s of a heart attack if you do not go on statins. And I'm talking even if you are a vegan marathon runner, you will still have problems. And if you're homozygous, so if it runs in your family, if you're homozygous, you have to be on statins from day one because you're likely to die in your 20s of a heart attack. Like I said, even if you're a marathon runner eating a plant-based diet, because your body can't remove the cholesterol from your blood, so it builds up in the plaque and arteries. So that's an example of incomplete dominance. Another one is plyotrophy. This is where one gene affects several traits. So we name all of our genes, right? They get all these different names. One is called the OCA2 gene, and it makes a protein needed to make melanin. Okay? So melanin is a pigment. That's what's in your skin. That's what's in your hair. That's what's in your eyes that make you dark. And the more melanin you have, the darker you are. And there's not just one type of melanin. There is different types of melanin, each produced by different types of genes. And then this OCA2 gene controls some of the melanin production. So we might have like an epistatic interaction, epistasis. This is where one gene controls the expression of another gene. So imagine you have a light and you've got a gene that acts as a switch that turns that light on and turns that light off. So in eye color, a lot of it is epistasis. You've got these different types of melanin. You have different controls on how much of that melanin you're producing. So you might have a gene to produce brown eyes, but then your other gene might not turn it on. So even though you've got the gene to produce brown eyes, it's turned off. It's like the light bulb doesn't get turned on. So you end up with light-colored eyes. So a mutation, for example, in the HERC2 gene. Now this is different from the one that's in breast cancer. That's just a HER2. Doesn't have the C in there. That's a gene that causes blue eyes because it's involved with the OCA2 gene in terms of turning it off so that if you don't have it on, you have blue eyes. So somewhere 6,000, 7,000, 8,000 years ago, my ancestors in Europe underwent a mutation to have blue eyes, and because it was desirable, it was kept in the population. Now, you could also have 2 or more genes involved with the trait. So this is a polygenic trait. Once again, eye color is a polygenic trait because it's not just one gene determining eye color. There are multiple genes involved with that. Skin color is another one. There are at least 3 different genes involved with producing melanin, and different types of melanin are slightly different color. So if you're of African descent, like my buddy here, this is one of my best friends, he's a workout partner. He's producing all kinds of melanin. I'm not producing as much. So this is a polygenic trait with multiple genes. So whether or not you're producing melanin is just a function of how much of your genes, right? And it's a protection against sunlight. So people in Africa live near the equator, so high amounts of melanin protected their skin from ultraviolet light. Whereas people in more northern latitudes, they didn't need all the protection. And by having less melanin, they produce more vitamin D. So that was a benefit. But the same mutation in Africa is bad, actually. And then there's phenotypic plasticity. I love King of the Hill. More Mike Judge, right? And also, for those of you that are interested in movies or TV, this guy also, not Mike Judge, but one of the other writers, also does The Office. Pretty cool. But phenotypic plasticity. That's just a fancy way of saying, okay, I've got a phenotype. That's your outward expression of your genes. Plasticity just means plastic. It can respond to the environment. Here's an example of phenotypic plasticity. Farmer's tan. All these guys have farmer's tans, right? Their arms are way darker than the rest of their body. What happens is your skin gets exposed to ultraviolet light, and your genes dictate how much more melanin you can produce to protect your skin from ultraviolet light. That's an example of phenotypic plasticity. If you run, you start making all kinds of physiological changes to your body to get in better shape. If you work out, you build more muscle to lift more weight. That is an example of phenotypic plasticity. Okay, now I'm going to finish out here really quick on the common misconceptions of genetics. 1, sexual reproduction does not create new alleles. It just shuffles them around. You're playing with your card deck. But mutations do make new alleles. Here's a good one. I hear this all the time. A dominant allele, like for a purple flower, it is not stronger. It's just expressing the trait, right? You're just making anthocyanin. The recessive one doesn't make anthocyanin. And it's not necessarily more common. And in fact, some dominant alleles might be very rare in a population, especially if they're bad. Genes don't exist to cause diseases. Even though we often discuss genes and the diseases they cause, those are mutations. Those genes aren't working. So one is achondroplasia. Here's, of course, Tyrion Lannister. He's got achondroplasia. His bones don't elongate. That is actually a dominant allele, but it's rare in a population because achondroplasia is bad. And if you've ever seen those little wiener dogs with those short little legs, that's achondroplasia. And it's actually bad. It's not good for the dogs to have that. I think it's kind of painful for them.